Good afternoon. I am Shoumi Sarkar, a biostatistics PhD student from the University of Florida. Today, I would be sharing our team's contribution to this year's AMR Forensics Challenge, which is about detecting bacteriophages associated with AMR when confounders are present. Before jumping into our objectives, I would like to introduce some terms. The first term is AMR, which is short for antimicrobial resistance. It occurs when microorganisms become immune to the very drugs used to inhibit them. The next term is resistomes or ARGs, which refer to the genes that encode for AMR. ARG is short for antimicrobial resistance genes. And next we have antibiotic classes, which are groups of drugs that inhibit the growth of microorganisms. They include groups of drugs such as penicillins, macrolides, beta-lactams, etc. Next we have phages, which are short for bacteriophages, that are viruses that attack bacteria and archaea. Next we have confounder, which is a statistical term for an underlying variable that interferes in the relationship between two other variables. For instance, the daily rate of drowning at sea is correlated with ice cream consumption. But would that mean that ice cream causes drowning? That is a very absurd idea. The truth is, on a sunny day, people are more likely to eat ice cream as well as go swimming. So temperature acts as a confounder. Temperature is a hidden variable that affects both ice cream consumption as well as the daily rate of drowning and causes both of them to increase if temperature increases and decrease if it decreases. And so it ends up affecting both these variables and making them seem as if one of them increases the other, where in fact that relationship does not exist. The main aim of our project is to study the role of phages in the spread of AMR by focusing on the relationship between phages and resistome abundances. AMR is a global health concern, and phages have been identified to spread resistant genes among bacteria by various mechanisms, such as horizontal gene transfer. However, the existing literature does not adequately explore this role. Some studies stress on their importance, while some others claim that they do not have a substantial impact. We use statistical and machine learning tools to assess the impact of phages alongside other established proponents of ARG dissemination on the spread of AMR. This would be a viable alternative to laboratory detection methods of finding important phages that have something to do with ARG spread. Here, we present the main steps of our analysis. After raw metagenomic samples are obtained in the data collection step, we pass them on to the preprocessing step, where we pass the reads through a standard bioinformatics pipeline to improve their quality. And once that's done, we compare the reads against databases to identify and count phages and resistomes in the taxonomic profiling stage. And finally, we are prepared to conduct our statistical analysis in the downstream analysis step. We now briefly go over the data collection step. Raw metagenomic sequence data from mass transit systems was provided by the CAMDA organizers. It was collected by the Metasub Consortium during Global City Sampling Days 2016 and 2017. We worked with 366 samples from several cities, both in and outside the US. Roughly half of the locations were inside the US and half of them were from other countries. Before we can extract counts of phages and resistomes from these reads, we need to preprocess these raw metagenomic sequences to improve their quality. In this step, we broadly removed human contamination, adapter sequences, and poor quality reads from raw sequences. The quality scores before and after preprocessing are shown here. On the left, we see that the quality scores were pretty low before preprocessing, but after, we see that the scores are pretty high and are acceptable for the further steps of our analysis. Next, we employed KMA to extract the counts, or abundances, of genetic information from these sequences. We downloaded the required reference sequences for phages and resistomes and built respective custom databases for referencing, and then mapped the preprocessed reads to these custom databases. This gave us matrices of resistome and phage abundances. This is how our abundances would look like. The rows are the samples and the blue columns are resistomes for 17 specific antibiotic classes. The green columns are for over 4,000 phages identified by their unique phage IDs. The data is high dimensional because we have more columns than rows, and sparse because our matrix has several zero values. The resistome abundances of 17 different antibiotic classes are our multivariate response. We have over 4,000 phages as explanatory or independent variables, many of which have zero counts. But we only have 366 samples in comparison. Alongside the genetic abundances, we also add some auxiliary information to enrich our dataset. While these are known to be associated with AMR, we suspect that they might potentially confound or affect the relationship between phages and resistomes. We introduced data on climate, demography, and landscape variables. We have 5 climate variables, including those on temperature and rainfall, as well as humidity. We also have variables on demography that include median annual household income, average age, population, and percentage of population with access to basic sanitation. And the landscape variables include those on air quality index or AQI, latitude, longitude, elevation above sea level, whether the city is a coastal city or not, and city land area. These were collected from various public databases from several national organizations. Here we present the layout of our data after we introduce the additional variables. In blue, we have our multivariate responses, the resistomes, across 17 different antibiotic classes. And then we have 4 blocks of independent variables, the blocks corresponding to phages, climate, demography, and landscape. These counts need to be pre-processed before we can start our analysis. For the phage abundances, we remove the phages with less than 5 reads, as well as those with presence in less than 1% of the samples. And then we normalize them using the metagenome-seq package in R. We also center and scale our auxiliary variables, and also employ the metagenome-seq package to normalize the counts of resistomes. The figure here summarizes the block structure in our data. We use the letters P to denote the number of variables and the letter K to denote the number of blocks. We have P1 variables on phages, P2 variables on climate, P3 variables on demography, and P4 variables on landscape. The 4 blocks, or the K blocks, explain our multivariate response. The figure here shows the distribution of resistomes over locations. We find that roughly the resistomes show similar abundances over different locations. We found that the 3 classes with the highest abundances of resistomes were macrolides, beta-lactams, and tetracyclines. Here, we present an overview of our analysis plan. With the preprocessed data, we try to realize 3 main objectives. We want to obtain block importances and variable importances to validate the need to prioritize phages, and then we take a closer look at the impact of phages on resistomes with potential confounders removed. Since our response Y is multivariate and our covariates X are high dimensional and arranged into blocks, it is very important that we select a model appropriate for data of this structure. So we choose to employ the multi-block partial least squares regression model to model Y on X. It tries to find the optimal linear combinations of independent variables that best explain the multivariate response while accounting for the block structure. We use 3 measures extracted from this model. One is BIPC for block importances, another is VIPC for overall variable importances, and PLS regression coefficients for variable importance specific to an antibiotic class. All of this could be obtained by using the package ADE4 in R. We employ a statistical tool called bootstrapping to obtain the 95% confidence intervals of the BIPC criteria. This helps us rank the 4 blocks on climate, demography, landscape, and phages to find which block is most important. We see that phages form the most significant block, whereas climate, demography, and landscape are not as important. When we looked at overall variable importance, we found that all our non-genetic factors or auxiliary variables turned out to be the most important. The rankings for these non-genetic factors agree with current literature. For instance, with regards to sanitation, it was found that improper disposal of fecal waste releases ARGs into the environment. As for coastal cities, we note that the proximity to water bodies and higher levels of human activity promotes the spread of ARGs. And also, a 2021 study explained the role of latitudes, finding more diversity of resistomes occurring near the equator. We have several other relationships which are further elucidated in our paper. After we listed the top 15 variables by VIPC, we list the next 100 variables. Interestingly, though phages appeared as the most important block, as variables they appeared only after the auxiliary variables. For optimal presentation, in this figure, we use phage IDs to denote the phages, but we present the corresponding phage names in this list. We see an abundance of Staphylococcus phages, and we find that the ones in blue would later match with the top phages identified at the antibiotic class levels as well. We now look at the top phages in the most abundant rhizostome groups. For macrolides, we find similar non-genetic variables at the top. Top phages in the previous overall list reappear at this class level list as well. With the beta-lactams antibiotic class, we find similar results with several phages in common with the previous lists. For the tetracyclines antibiotic class, we find similar results as well, with similar auxiliary variables selected as the top ones, followed by top phages that correspond to those found in the previous class-specific as well as overall lists. We have some very interesting observations. The block of phages turns out to be the most important. However, at the variable level, the non-genetic factors come first. It is very possible that the importance of phages is somehow masked or absorbed by the auxiliary variables. If they did not interfere, would the relationship look any different? Another observation is that the Staphylococcus phages dominate our top list of phages, but only 4% of all phages are Staphylococcus phages. It is really concerning that no other categories of phages are that highly associated with AMR. To address these concerns, we adjust for these possible confounders so that they no longer interfere between the relationship between phages and rhizostomes. With the notation YR for rhizostomes in the R antibiotic class, where R varies from 1 through 17, we utilize a two-step strategy. For each class, we fit the rhizostomes on auxiliary variables, or the non-genetic factors, and then we obtain residuals YR star, which correspond to rhizostome abundances with the effect of the non-genetic factors removed. And then we employ random forests to model these class-specific residuals on phages to get importance scores. However, this would give us 17 class-specific importances with phages in possibly different orderings of importance. So to arrive at a consensus, we use rank aggregation, which combines the scores in multiple lists into a single ordered top K list, for instance, a top 20 or a top 30 list. The importance scores from random forest are used as weights. We obtain the following top 20 list from rank aggregation. The list is quite different from the initial list obtained from VIPC scores. While the phages in blue are the ones that match with the previous list, we find that Staphylococcus phages no longer dominate this list, and phages that attack other kinds of bacteria are also recognized. The top phages selected here agree with those established in clinical literature. For instance, the phage RCS47 was found to contain a certain kind of beta-lactamase that is a class of enzymes that are produced by bacteria to break open the beta-lactam ring of the beta-lactam antibiotic, rendering it inactive. Next, a certain Staphylococcus phage was found to carry a gene resistant to the antibiotic chloramphenicol. These are just examples, and we have several other findings elaborated in our paper. To ensure that these phages were not selected by random chance, we looked at the probabilities of their inclusion in the top 20 list and found that these quantities were well above the corresponding threshold of 0.0168. The threshold was computed combining ideas of jackknifing and probability theory. From the 366 samples, we left one sample out and repeated the steps to obtain the corresponding top 20 rank aggregated list, and repeated this for all 366 samples to get 366 lists. We then find the proportion of phages in the original shortlist, the one where no samples were removed, that occurred in these 366 lists. We call these the inclusion probabilities for each phage. The threshold is computed using a hypergeometric distribution, which looks at the sampling without replacement from two groups in the population. Here, the 20 phages in the original top 20 list forms the first group, and the remaining 1,170 phages form the second group. This leads to the threshold of 0.0168. To conclude, we explored a new approach of identifying the primary AMR drivers with a focus on phages. While non-genetic factors play an important role, their importance is already established in literature. But ranking blocks revealed that phages needed a closer look. Overall, variable importances suggested that confounding effects might be at play. After obtaining an updated list of top phages after adjusting for possible confounders, we found meaningful results with an agreement between these phages and clinical literature. I would like to acknowledge my team, my colleague and collaborator Sam, and our advisor, Dr. Somnath Datta at the University of Florida. Here is a list of references for our project. And thank you for your attention.